Good morning, class. Today's topic is calcium homeostasis. So, calcium and phosphate homeostasis is discussed under two separate classes. So, this is the first part. In this class, we will discuss the calcium and phosphate basic metabolism, how they are absorbed and reabsorbed, and how the constancy of the internal environment, that is homeostasis, is maintained for their levels, that is calcium and phosphate levels in the external cellular fluid, that is ECF. Okay, so the learning objectives for today's topic are calcium and phosphate metabolisms, the functions of calcium and phosphate in the body, and various aspects of, that is the basics of bone physiology and adult bone remodeling and physiological regulation of calcium and phosphate homeostasis. So this calcium and phosphate metabolism you already had in your biochemistry class, but again for physiology just we will discuss few basic points. Calcium is the most abundant mineral of our body. The total body calcium is 1100 to 1200 grams or 1.5% of body weight. So out of which 99% is present in the skeleton, 4 to 5 grams in soft tissue and only 1 gram in extracellular fluid. So this 1 gram is called normal serum calcium. The value of normal serum calcium is 8.8 to 10.4 milligram percentage, or you can say that 9 to 11 milligram percentage. So the normal serum calcium is 9 to 11 milligram percentage, and this value is very important. It will be asked as MCQ or any in Vivas, or also when you become a physician you should check the normal values. So this normal serum calcium value is very important, please remember. So this plasma calcium occurs in two forms, diffusible and non-diffusible. So diffusible form is 5.36 milligram percentage or 54 to 55 percentage, and which again is divided into ionized 47 and non-ionized 5%. And non-diffusible form occurs to 45 to 46 percentage or 4.64 milligram percentage. So this tight range, that is 1 to 2 gram percentage variation, it is very important. It shows that its importance in maintaining the critical body functions, and it's always regulated in this range of 9 to 11 milligram percentage. So before the 5th month of intrauterine life, very little calcium is found in fetus because bone formation is only starting stage. So before the 5th month of intrauterine life, very little calcium is found in fetus. And 60% of total deposition occurs in the last trimester, that is 7th, 8th and 9th months, last 12 weeks, which is a period of rapid and extensive ossification. So in that time period, 60% of total deposition. So not complete 100%, it's only 60% of total deposition occurs in the last trimester. And before 5th month, only very little calcium is found in the fetus. So after birth, the process of bone complete remodeling or the bone ossification process takes place as the child grows. So this is the picture of calcium metabolism. So from milk, cheese, eggs and hard water, that is the sources of calcium, we get calcium about, here roughly we have taken the values 20 millimole per day or 12 to 35 millimoles per day. 1 millimole of calcium is equal to 40 mg of calcium. So in the intestine, the calcium is absorbed. So 30% of calcium is generally absorbed and 70% is excreted in stool or feces. And 150 grams of milligrams of calcium is secreted into the intestine. Or here we have shown 18 millimoles per day. So that amounts to roughly 720 milligrams of calcium, or nearly 1000 milligrams of calcium is excreted daily from the body. So after absorption from the intestine, it enters the extracellular fluid. So this is in equilibrium with the rapidly exchangeable pool of the body. So from the rapidly exchangeable pool of plasma, 500 milligrams of calcium enters the bones, and then for which is used for bone remodeling and bone formation, and about 500 milligrams of calcium is again extruded back into the plasma from the bones. And from the urine, about 150 milligrams, or here 2 millimoles per day, that is for 80 mg of calcium is excreted in urine. So there is increased calcium requirements during pregnancy and nursing. The absorption and excretion of calcium in the body, 35 to 40% of daily dietary calcium is absorbed from the gut, and remaining 70% or 60 to 70% is excreted in the feces. So absorption is mainly from duodenum and first half of jejunum by carrier mediated active transport under the influence of vitamin D. So after oral administration, that is when we take the calcium externally, absorption is completed within 4 hours. So we will see the factors affecting calcium absorption from the GIT. So increased by acidity in the stomach, calcium phosphate ratio, hypocalcemia during pregnancy and lactation, vitamin D3. Under the influence of vitamin D3, calcium absorption is increased, and also parathyroid hormone. So these two are very, very important hormones in regulating the calcium homeostasis, and lactose, and decreased by intestinal alkalinity, that is opposite for this. Acidity causes increased absorption, alkalinity causes decreased absorption, and excess of oxalate and phytic acids, and hypercalcemia, and also lipids or fats in the diet decreases the absorption of calcium. Alcohol and smoking and lack of exercise decreases absorption. Emotional stability and glucocorticoids decrease the absorption of calcium from GIT. Coming to the excretion of calcium. So calcium is both filtered and reabsorbed but not secreted. The rate of renal calcium excretion is calculated as calcium filtered minus calcium reabsorbed. So that will be the calcium excretion. 99% of filtered calcium from glomerulus is reabsorbed by the tubules, and only 1% gets excreted. 65% is reabsorbed in proximal tubules, and 25 to 30% in loop of Henle, 4 to 9% in distal and collecting tubules. So that accounts to 99% of filtered calcium. So that is reabsorbed in parts. Proximal tubules 65%, loop of Henle 25 to 30, distal and collecting tubules 4 to 9%. And daily loss of calcium in the sweat is about 15 milligrams. So this is the picture showing calcium excretion in feces, that is 18 millimole per day or 720 milligrams of calcium, and in urine 2 millimoles per day. Coming to the functions of calcium. Calcium is very important ion in the body which is tightly regulated in the range of 1 to 2 milligram percentage, and majority of which is present in the bone, that is skeleton. So the varied physiological functions which involve calcium are bone and teeth formation, and neuronal activity like genesis and maintenance of action potentials, and genesis of pacemaker potentials, and also neurotransmitter released from the nerve terminals. And muscle activities, you already know that excitation contraction coupling takes place in the muscle, and for contraction this calcium storage from the endoplasmic reticulum, calcium release from the endoplasmic reticulum is very, very important for the contraction to take place. And cardiac activity, that is pacemaker potential, and also the contraction of cardiac muscle is also maintained, or it requires the calcium presence. And cell division and growth requires calcium. And blood coagulation, we all know that factor 4 itself is calcium, so it triggers or it helps in the maintenance of cascade of blood coagulation. without which the coagulation process does not take place. And excitability of nerve and muscle requires calcium because it influences the sodium permeability and it influences the ease with which the action potentials are triggered and maintains the integrity of cell membrane. And it acts as a second messenger or third messenger in various extracellular, intracellular signaling pathways for hormone actions. Coming to phosphorus, which is a key element in all the known forms of life. It plays a major role in biological molecules such as the genetic material RNA and DNA, and it is a main structural component of all the cellular membranes. And living cells also utilize phosphate to transport cellular energy via ATP, adenosine triphosphate. And an average person contains little less than 1 kg of phosphorus, about 3/4 present in bones and teeth in the form of aptite crystal. So if we see the phosphate metabolism, phosphate is found in ATP, cyclic AMP, 2,3-DPG and cofactors like NAD and ADP. And the total body phosphate is 500 to 600 grams, out of which 80 to 85% is present in skeleton, whereas the calcium 90 to 99% is present in skeleton. And the remaining phosphate 10 to 15% is present in intracellular phosphate pool. And the serum inorganic phosphate level in adults is 2.5 to 4 milligram percentage and in children it is 5 to 6 milligram percentage. So this is the picture showing the phosphate metabolism. So the daily phosphate intake is about 1.4 gram per day, out of which 75% is absorbed from the intestine. Okay, so from the intestine 75% is absorbed and 25% is excreted in the stools. The phosphate pool in ECF is about 700 mg, from where about 200 mg is excreted with the bones for bone remodeling. So the phosphate pool in the ECF is 700 mg, from where about 200 milligrams is exchanged with the bones for bone remodeling. And normally 1000 milligram of phosphate is excreted in urine, that is 1 gram per day. And fecal excretion is 400 milligram per day. So total excretion is 1.4 gram per day. And regulation of renal excretion of phosphate mainly controls the concentration of plasma phosphate. Phosphate deficiency results in muscle weakness, cardiac and respiratory dysfunction, abnormal bone formation, and loss of red cell membrane integrity. Okay, so here the values to remember: dietary intake 1.4 gram per day, and exactly the same is excreted out of the body via feces and urinary. So urinary excretion is a major part. So reabsorption and filtration from the ECF and kidney, it is 6 gram per day. Reabsorption 5 gram, filtration is 6 gram, and from GIT absorption is 1200 milligram per day and secretion is 200 milligram per day. So ECF value is 700 milligram, out of which 200 is deposited and reabsorbed from the bones, 200 milligram per day. So this is the metabolism of phosphorus. Coming to the distribution of phosphate, so total phosphate 500 to 800 milligram, out of which 80 to 85% is present in bones and teeth, and normal plasma levels are 2.5 to 4.5 milligram per deciliter, out of which organic accounts to 0.5 to 1 mg per deciliter, inorganic in adults 3 to 4, and children 5 to 6 mg per deciliter. So as I have told you, the value of serum calcium, which is very important, that is 9 to 11 milligram percentage. So likewise serum phosphate levels are also important, you should remember, that is 2.5 to 4.5, or else you can remember this inorganic phosphate value, that is 3 to 4 milligram per deciliter. And next is the phosphate absorption. The mechanism of phosphate absorption is via co-transporter, that is sodium phosphate 2B, so which is related to transporter in kidney. Okay, so it is co-transported with sodium. So phosphate is absorbed in duodenum and other parts of small intestine by active transport and passive diffusion. And the daily requirement of phosphorus varies in adults and children. So in children it is high 800 to 1200 milligram per day, and in infants it is very less 240 to 400 milligrams, and in adults it is 800 milligram per day, and also in pregnancy and lactation the value increases to 1200 milligram per day. And phosphorus is excreted in urine and feces. So in majority in urine 1 gram per day, and feces only 400 milligram per day. Urine phosphate constitutes about 60% of total excretion, and the rest is excreted in feces. So the functions of phosphorus are very less compared to calcium. So like for the calcium, it is also present in bone and teeth. So 80 to 85% of phosphorus is present in skeleton, and it is a structural component of all the cells, and it is involved in the intermediate metabolism because it is required for phosphorylation of many intracellular proteins for the formation of phosphoproteins. It acts as a covalent modifier for many enzymes, and it is the genetic material, that is RNA and DNA. It is the component of RNA and DNA, and it serves as an important component of intracellular pH buffering system. Coming to the physiology of bone, bone is a well vascularized living connective tissue, and it plays very important role in calcium and phosphate homeostasis in addition to its other functions. So it is composed of tough organic matrix that is greatly strengthened by deposits of calcium salts. Average compact bone contains by weight about 30% matrix and 70% salts. So this organic matrix include collagen fibers, which accounts to 90 to 95%, and ground substance, which is 5 to 10%. Ground substance is again composed of chondroitin sulfate and hyaluronic acid. And if we see the histology of bone, like other connective tissues, it also contains abundant matrix surrounding the cells. The matrix is about 25% water, 25% protein fibers, and 50% mineral salts. And there are 4 types of cells in the bone tissue: osteoprogenitor cells, and osteoblasts, osteoclasts, osteocytes. So you all know about these types of cells in your intermediate, but again here just a brief description about those cells. So this osteoprogenitor cells, progenitor means the first main before the cells which are present before the osteoblasts, osteocytes, and osteoclasts. So these are unspecialized cells derived from mesenchyme, the tissue from which all the connective tissues are derived. They undergo mitosis and develop into osteoblasts. They are found in the periosteum, endosteum, and in canals that contain blood vessels. So they undergo mitosis and develop into osteoblasts. So these are the before cells. Pro means before. For any type of cell, if the word pro comes, it is before. And osteoblasts are the cells that form bone, but they have lost the ability to divide by mitosis. So progenitor cells have the ability to divide by mitosis, and osteoblasts will be losing that capacity. They secrete the collagen and other organic components needed to build bone tissue. The differentiation of osteoprogenitor cells into osteoblasts is accelerated by some hormones and some bone proteins called skeletal growth factors. And the function of these osteoblasts are formation of bone matrix and calcification through alkaline phosphatase enzymes and synthesis of proteins. And the osteocytes are the cells concerned with maintenance of bone tissue, small, flattened and round cells embedded in the bone lacunae. And these are derived from mature osteoblasts. Okay, so osteocytes are nothing but derivation of osteoblast cells, and which are flattened, rounded cells embedded in the bone lacunae. So if you can see here, these are the progenitor cells which develop into osteoblasts, which form the matrix of bone tissue, and some osteoblasts will differentiate into osteocyte. These cells' function is to maintain the bone as living tissue because of their metabolic activity, and it helps in maintaining the exchange of calcium between the bone and extracellular fluid. And osteoclasts are concerned with bone resorption. Okay, so bone absorption of calcium from bone, and resorption is From the bone, the calcium is being again taken back into the extracellular fluid, that is resorption. Giant phagocytic multinucleated cell found in the lacunae of bone matrix. So this is the yellow colored one, that is osteoclast. It is derived from hematopoietic stem cells via monocytes. And the function is responsible for the bone resorption during bone remodeling. And synthesis and release of lysosomal enzymes necessary for bone resorption into bone resorbing compartment. So synthesis and release of these lysosomal enzymes which are necessary for the bone resorption into bone resorbing compartment. So we have lamellar and woven type of bones. So lamellar means lamellae. They are placed in layers. When we examine the structure of any adult bone, we find that it is made up of layers or lamellae. This kind of bone is lamellar bone. So in the picture you can see the layers of bone. So between adjoining lamellae there are spaces called lacunae. These spaces are occupied by cells of bone, that is osteocytes. And woven bone is the newly formed bone which does not have lamellae. So that is lamellar bone is the adult mature bone. And this woven bone is a newly formed one which does not have lamellar structure. And the bundles of collagen fibers run randomly and interlace with each other. And because of that it is called woven. So interlacing the bundles of collagen fibers which interlace with each other are present in that woven bone. All newly formed bones are woven bones and it is later replaced by lamellar bone. And next, compact or cortical bone. This type of bone is made up of lamellae which are arranged in the form of concentric rings that surround a narrow Haversian canal present at the center of each ring. So the bone which is made up of concentric rings which surround a narrow Haversian canal which is present at the center of each ring. The blood vessels, lymphatic vessels and nerves from the periosteum penetrate the compact bone through perforating or Volkmann's canals. The blood vessels and nerves of these canals connect with the blood vessels and nerves of medullary cavity and periosteum and those of the central Haversian canal. Okay. So all this anatomy of bone you already had in your starting classes of anatomy, that is skeletal system. So again this will be just a brief revision like thing for you. So here you can see the structure of a compact bone and spongy bone. So this is the trabecular spongy bone and these are the osteons of compact bone. So in the compact bone you have the Haversian canal and Volkmann's canal. So and here is also you can see the chondritic lamellae and interstitial lamellae. So one Haversian canal and the lamellae around it constitute a Haversian system or osteon. The central canals run longitudinally through the bone. Around the canals are concentric lamellae. And the area between osteons contain interstitial lamellae. These have lacunae with osteocytes and canaliculi, but their laminae are usually not connected to osteons. So spongy or trabecular or cancellous bone. So in contrast to compact, this does not contain true osteons. Instead, it contains lamellae arranged in an irregular lattice work or network of thin plates of bone called trabeculae. So in the previous slide, you have seen the central part, trabeculae. There are thin plates of bone arranged in a network or lattice work. So that is why it is called trabecular bone. And the spaces between the trabeculae of some bones are filled with red marrow that will produce blood cells. So the central part of bone is made up of red marrow in the spongy bone, which produces the red blood cells. In the physiology of bone formation, that is ossification. The process of bone formation is called ossification. The skeleton of human embryo is composed of fibrous connective tissue membrane formed by embryonic connective tissue, mesenchyme and hyaline cartilage that are loosely shaped like bones. They provide supporting structure for ossification. Ossification begins around 6th or 7th week of embryonic life and continues throughout adulthood. Bone formation follows one of the two patterns, intramembranous ossification and endochondral ossification. Intramembranous refers to formation of bone directly on or within the fibrous connective tissue membranes. And endochondral refers to formation of bone in hyaline cartilage. So intramembranous is within the fibrous connective tissue membrane, and endochondral is hyaline cartilage, formation of bone in hyaline cartilage. Maxilla, for example, maxilla forms by intramembranous ossification, and mandible forms partly by intramembranous and partly by intracartilaginous or endochondral. And greater part of the body, ramus, condyloid, and coronoid process of mandible are intramembranous. Only the tip of condyloid and coronoid process are of endochondral origin. If we see the intramembranous ossification, it is at the site where bone will develop. Mesenchymal cells become vascularized, cluster and differentiate first into osteoprogenitor cells and then into osteoblasts. The site of such a cluster is called center of ossification. Osteoblasts secrete the organic matrix of bone and get surrounded to become osteocytes. Later, calcium and other minerals are deposited and the tissue calcifies. So, this is the process of intramembranous ossification. First, the center of ossification develops. So, from there, osteoblasts will secrete the organic matrix and get surrounded to become the osteocytes. And later, calcium and other minerals are deposited so that the tissue calcifies. As the bone matrix forms, it develops into trabeculae. As the trabeculae develop in various ossification centers, they fuse with one another to create the open lattice network appearance of spongy bone. Connective tissue in trabecular spaces differentiates into red bone marrow. On the outside of bone, vascularized mesenchyme develops into periosteum. Eventually, some of the spongy bone is replaced by cortical bone. This will be remodeled to reach its adult size and shape. So first, the center will be formed and the osteoblasts, osteocytes, and the deposition of calcium and other minerals, and then it develops into trabeculae, and then the spongy bone and the outer area outside of the bone will be developing into periosteum, and some of the spongy bone will be replaced by cortical, and this will be remodeled to reach the adult size and shape. That is the process of entire bone formation. So in pictorial form you can see here an ossification center appears in the fibrous connective tissue membrane. So mesenchymal cell, collagen fibers, ossification center, and these are the osteoid, osteoblast cells. And then the bone matrix, osteoid is secreted within the fibrous membrane. Osteoblasts begin to secrete osteoid, which is mineralized within a few days. Trapped osteoblasts become osteocytes. And then the woven bone and periosteum formation occurs. Accumulating osteoid is laid down between the embryonic blood vessels, which form a random network. The result is network instead of lamella of trabeculae. That is, the bone will be arranged in the form of a thin plates of bone arranged in the form of a trabecula. And the vascularized mesenchyme condenses on the external face of the woven bone and becomes the periosteum. So here you can see the outside of the bone, mesenchyme condensing to form the periosteum. And bone collar of compact bone forms and red marrow appears. So trabeculae just deep to the periosteum thicken. So here this trabeculae just deep to the periosteum thicken, forming a woven bone collar that is later replaced with mature granular bone. Spongy bone dipole consists of distinct trabeculae, persists internally, and its vascular tissue becomes red marrow. So this vascular tissue, the center part of the bone, will become red marrow. And then the endochondral ossification, it begins in second month of development. It uses hyaline cartilage bones as models for bone construction. It requires breakdown of hyaline cartilage prior to ossification. So first, development of cartilage model. Mesenchymal cells differentiate into chondroblasts, which form the hyaline cartilage model. A membrane called perichondrium develops around the cartilage. And then this cartilage model grows by interstitial and oppositional growth. Chondrocytes in the mid-region calcify the matrix, and the vacated lacunae forms small cavities, and the osteoblasts in perichondrium produce periosteal bone collar. Once perichondrium starts to form bone, it is known as periosteum. So in the picture you can see the central part, it is becoming calcified, and the adjacent outer part is the periosteal bone collar.
Development of primary ossification center. Near the middle of the model, capillaries of periosteum grow into disintegrating calcified cartilage, and these vessels and osteoclasts, and adenoid cells are known as periosteal bud. With the development of periosteal bud, primary ossification center and medullary cavity forms.
So you see primary ossification center with periosteal bud and medullary cavity appearing here, and the nutrient artery enters the bone. So this is the calcified matrix and uncalcified matrix. This part is pericorneum because it's not still uh, modified. So the modified pericardium will become the periosteum. And the development of diaphysis and epiphysis. The diaphysis, which was once a solid mass of hyaline cartilage, is replaced by compact bone. When the blood vessels, epiphyseal arteries enter the epiphysis, secondary ossification centers develop, usually around the time of birth. So secondary ossification centers develop at the epiphysis. So diaphysis is the central long bone, the central part. The epiphysis are the edges, ends of the bone. So till now we have seen the various types of cells and ossification of the bone, and now we will see the physiology of bone growth. So bone grows in length at the epiphyseal plate by a process that is similar to endochondral ossification. The cartilage in the region of epiphyseal plate next to epiphysis continues to grow by mitosis. So epiphyseal plate next to epiphysis, so that in that region the cartilage which is present that continues to grow, and the chondrocytes in the region next to diaphysis age and degenerate, that is they will be destroyed, and the osteoblasts will be moving in the place of chondrocytes and ossify the matrix to form bone. So this process continues throughout the childhood and the adolescent years, that is until 17-18, and then until the cartilage growth slows down and finally stops. So till that point, this process of replacement of chondrocytes with osteoblasts continues to occur, and the epiphyseal plate completely ossifies so that only a thin epiphyseal line remains and the bone can no longer grow in length. Even though bones stop growing in length in early adulthood, they can continue to increase in thickness or diameter throughout life in response to stress from increased muscle activity or to weight. So we all know that the growth in length stops in early adulthood, but the thickness and diameter increases throughout life in response to stress from increased muscle activity or when the weight of the person increases. So that is called appositional growth. And the osteoblasts in the periosteum form compact bone around the external bone surface. Osteoblasts. At the same time, osteoclasts in the endosteum break down bone on the internal bone surface around the medullary cavity. So these two processes by osteoblasts and osteoclasts help in increasing the diameter of bone. And next we will see the bone homeostasis. Bone, like skin, forms before birth but continually renews itself thereafter. So bone and skin, both the structures form before birth only, but they continually renew. So the cells will be degenerated and new cells will be produced throughout the life. Remodeling is the ongoing replacement of old bone tissue by new bone tissue. It takes place at different rates in various parts of the body. Osteoclasts are responsible for bone resorption, that is destruction of matrix. A delicate homeostasis exists between the actions of osteoclasts in removing minerals and collagen and of osteoblasts in depositing them. So osteoblasts will deposit the minerals into the bone and osteoclasts will be removing the minerals and collagen from the bone. So that is resorption, destruction of matrix. A loss of too much calcium or tissue weakens the bones and they break as occurs in osteoporosis or become too flexible as in osteomalacia. Abnormal acceleration of remodeling process results in a condition called Paget's disease. So the ARF cycle of bone remodeling is: A is activation. Osteoclasts are activated and begin secreting acids to resorb bone, and resorption. R is resorption and reversal. Resorption is osteoclastic resorption occurs and reversal is resorption stops and osteoblasts take over, and formation is osteoblasts form bone on the opposing surface to complete the bone reforming process. So this cycle takes about 100 days in compact bone and 200 days in spongy bone. Activation, resorption, reversal and then formation. So this is the out-ref cycle of bone remodeling. So first, bone adjusts its strength in proportional to degree of bone stress. So the bones thicken when subjected to heavy loads. And second, even the shape of bone can be rearranged for proper support of mechanical forces in accordance with stress patterns. So the shape of the bone can also be rearranged or deformed for support of mechanical forces in accordance with the stress patterns. So depending on the person uh, in which profession he is, if he is a weightlifter or something like that, the shape of the bone can also be rearranged. And thirdly, new organic matrix is needed as the old organic matrix degenerates. So long bone growth and remodeling, if you can see here, this is the growth and this side it is remodeling. So bone grows in length because cartilage grows in the edges and then replaced by bone in the inside, and here on the horizontal side you can see cartilage grows inside and then replaced by bone outside this side. So these two are the differences. So remodeling, growing shaft is remodeled by bone resorption and then bone added by oppositional growth and bone resorption again from the inside. So first here from outside bone resorption starts and then bone added by an oppositional growth in the that is growing in diameter, and then from inside again bone resorption occurs. So that is remodeling, continuous osteoclastic resorption and then addition of bone and then again resorption. So that ARF cycle continues throughout the life. Minerals needed for bone remodeling. Normal bone growth in young and bone replacement in the adult depend on presence of several minerals. Sufficient amount of calcium and phosphorus, component of hydroxyapatite. The primary salts that make the bone matrix hard must be included in the diet. So these are the primary salts, calcium and phosphorus, which must be included in the diet. Magnesium deficiency inhibits the activity of osteoblasts, and boron, which helps in bone growth. Manganese deficiency inhibits laying down of new bone tissue. Vitamins needed for bone remodeling like are vitamin D, C, A, B12. The most active form of vitamin D is calcitriol. Acting as a hormone, it promotes removal of calcium from the bone. On the other hand, it retards calcium loss in urine, which makes it available for deposition in bone matrix. And the vitamin C deficiency causes decreased collagen production, which retards bone growth and delays fracture healing. Vitamin A helps to control the activity, distribution, and coordination of osteoblasts and osteoclasts during development. Its deficiency results in decreased rate of growth in skeleton. Vitamin B12 play a role in osteoblast activity. So vitamin D is the main hormone. The active form is calcitriol, and vitamin C helps in the collagen production, and vitamin A controls the activity, distribution and coordination of osteoblasts and clasts during development. And B12 also has role in osteoblast activity. And the hormones that affect homeostasis of bone tissue and remodeling. Human growth hormone. Its function is general growth of all body tissues including bone. And sex hormones increases bone building activity of osteoblasts. Insulin and thyroid hormones promote normal bone growth and maturity. Parathyroid hormone increases the number and activity of osteoclasts, promotes recovery of calcium from urine and promotes formation of calcitriol. Calcitonin inhibits the activity of osteoclasts, speed up calcium absorption from blood and accelerates calcium deposition by bones. So the hormonal regulation of bone growth during youth. During infancy and childhood, epiphyseal plate activity is stimulated by growth hormone, and during puberty, sex hormones initially promote adolescent growth spurts and then cause masculinization and feminization of specific parts of skeleton, and later induce epiphyseal plate closure, ending longitudinal bone growth. So till now we have seen the bone physiology, the bone formation or ossification, and then bone remodeling. And then we have seen in the first slides the calcium and phosphorus metabolisms and their daily dietary intake and distribution in the body and their absorption, excretion, all that we have seen. So now the last part of today's class, the calcium and phosphate homeostasis, or how these calcium and phosphate are being regulated at their normal values in the body. So homeostasis is influenced mainly by the exchange of these calcium and phosphate between the plasma and the bone. Absorption of calcium and phosphate from intestine and kidney that determine their fecal and urinary excretion respectively greatly contributes to their homeostasis. So in general, calcium is excreted more in the stool and less in urine, whereas phosphate is excreted more in urine and less in stool. If you have remembered from the first starting slides, so calcium is excreted more in the stool and phosphate is excreted more in urine. And this homeostasis is having two controls, two types of controls. One is intestinal and other is renal. So intestine and kidney are the main controlling organs for the calcium and phosphate. Intestinal control, absorption of calcium and phosphate from intestine significantly contributes to their plasma concentration. And renal reabsorption of calcium and phosphate from the kidney significantly contributes to their plasma concentration. So the serum calcium normal values are 9 to 11 mg per deciliter and phosphorus 3 to 4.5 mg per deciliter. These two values you have to remember. So the relationship is always maintained. When the calcium is increased, phosphorus will decrease. When the calcium is decreased, phosphorus will increase. So this calcium and phosphorus homeostasis is maintained continuously by the intestine and kidney with the help of hormones like parathyroid, vitamin D, and then calcitonin. So, what are those hormones? How they regulate this calcium and phosphorus homeostasis? We will see in the next class. So, factors affecting the blood calcium concentrations are, so as I have told you, parathormone, calcitriol or vitamin D, and then calcitonin. So, this will be raising the level of calcium in the blood. Okay, and then the organs are gut, bone, and kidney. So, bone again, there will be resorption and absorption of calcium. From the intestine, calcium is absorbed, and from the kidney, calcium is reabsorbed. From the blood, calcium will enter into the kidney and again reabsorbed. So this picture shows a condition when the plasma phosphate is decreased. So because the calcium and phosphorus are inversely related, their values are inversely related. So there will be increased plasma calcium which will inhibit the parathyroid glands so that parathormone level decreases, that will cause increased phosphate resorption by the kidneys and decreased calcium reabsorption by kidneys, so that calcium is more excreted in urine and calcium is less excreted in urine. So that will lead to increased plasma phosphate, that is the value will be becoming normal. First is decreased plasma, so that will be feedback, with the help of feedback mechanisms it will be regained to normal values, and on the other side the kidneys will be stimulated, which will produce activated vitamin D that will cause increased calcium absorption from intestine. So there will be no change in plasma because of increased urinary excretion of calcium and the increased absorption from the intestine. So those two will be contracting each other, so there is no change in plasma calcium. And here on this side, there will be increased phosphate absorption in the intestine, leading to increased plasma phosphate concentration. So this is the picture of regulation of phosphorus concentration, or the value of normal serum plasma phosphorus. So that's the end of today's class. We have seen the calcium and phosphorus metabolism, bone physiology, and then the calcium and phosphorus homeostasis, how they are regulated at their normal values. So from this class, we will be getting the MCQs, and for knowledge process, all the bone physiology we have seen with many slides, and calcium and phosphate metabolisms and their homeostasis, how they are regulated at the normal values. So these may not come as say question or short notes in your exams, but these are important for Viva and MCQs. So don't neglect, listen carefully. If you don't understand anything, message me. So for tomorrow class, the important part of the calcium and phosphate homeostasis, that is, we will see all the hormones, parathyroid hormone, vitamin D and calcitonin, which will be helpful in the regulation of calcium and phosphate homeostasis. So thank you all.